Ladies and gentlemen, welcome back to the second session of the morning. To highlight the importance of metrology in the life science, we will hear presentations from two dynamically developing areas of medical research. Our first speaker is Renée Ruhaak. She is an associate professor at the Department of Clinical Chemistry and Laboratory Medicine at Leiden University Medical Center. She has a background in analytical chemistry, specifically focusing on proteins and post-translational modifications. Her research focuses on both development and implementation of quantitative protein mass spectrometry tests, as well as the role of mass spectrometry in metrology and test standardization. Ultimately, her goal is to contribute to a more sustainable healthcare system through the enabling of P5 medicine by clinical proteoform testing, where P5 stands for personalized, predictive, participatory, precision, and preventive medicine. Renée, we are excited to learn more about personalized medicine for improved patient care. Welcome to the stage.
Thank you for introducing me, and I would also like to thank the organizers for giving me the opportunity to present the work that we are doing within our laboratory. So I am at the moment an associate professor within the Department of Clinical Chemistry and Laboratory Medicine at the Leiden University Medical Center, and I'm heading mostly the research and development team of this unit, which is kind of split between two major tasks or major things that we do. One is the development of novel tests, where we really try to advance laboratory medicine through novel biomarkers and novel targets. But on the other hand, we also spend quite a lot of time on aiming for global standardization. And today I would like to present basically where these two come together. So I really appreciate being here, and I really appreciate being able also to speak to you. So like I said, like the title of my presentation, I'm going to speak about personalized medicine for improved patient care, and specifically the role of mass spectrometry-based precision diagnostics with a focus on coagulation. An overview of my talk is that I would like to first give a little bit of an introduction of metrology in clinical chemistry and how it is organized, then go towards precision medicine and the role of proteins and metrology. So what really, within our laboratory, within our clinical chemistry laboratory, we aim to add a value to healthcare through clinically effective medical tests, and what we basically do is that more or less any of the bodily fluids that you can basically take from an individual, we measure compounds in these, and we try to aid in diagnosis. And approximately 70% of the medical decisions that are being made, not only in a hospital, but generally I would say, is guided by clinical chemistry tests. Within 1 hour, clinical test results may provide life-saving information. There are now, for instance, pilots running where we even, within the ambulance, are already measuring troponins, and we're trying to triage whether an individual actually has to come to the hospital for cardiac, basically for heart attacks, or whether the results are in such a way that that it is necessary. So there really are quite a lot of medical implications that are depending on the medical tests that we perform. So the aim of standardization of medical tests is to provide consistent results no matter what position in time and space. So basically, if I go to a hospital here in Paris, I would like to get the same result as whether I go to a hospital in the Netherlands. And specifically, this is prerequisite to have global decision limits. So basically, we should be able to to define decision limit and use them globally to also have global reference values and to have global treatment targets. And why I think this is very important is because doing these studies to define decision limits, reference values, and specifically also treatment targets, these are very costly randomized controlled trials most of the time. So there is a lot of money that is involved, and it would be really a pity if we would have to redo this all over again for every, say, manufacturer of a test again. So in clinical chemistry, the standardization starts with the availability of a complete metrological traceability chain, which is described in ISO 17511. And I think that you are quite aware of these metrological traceability chains. But what I think is rather important is that there is on top here, it's mostly metrology institutes that are kind of providing the top of the traceability chain. Then we generally have a layer of IVD manufacturers who use this top of the metrology chain to calibrate. And the lowest part, actually here at the bottom, is where the medical laboratories find their place and where they actually apply those kits that are brought on the market by the IVD industry. So standardization of clinical tests is generally, I would say, influenced or maybe even in part decided by the International Federation of Clinical Chemistry and Laboratory Medicine, and their mission is to advance the science of clinical chemistry and to apply it to the practice of clinical laboratory medicine. And I think what is really important is that they are generally governors of what type of standardization projects are initiated. Currently, they have an executive committee, 6 team-oriented committees, and 20 working groups, and these are task-oriented, and mostly within working groups, that is where the standardization projects are actually taking place. So, what the IFCC does, and I'm going to just get everything in, is how do they decide on what type of projects to actually do? They either do horizon scanning, where they say, okay, what is important for us to do, and there's also what they call third-party approach, where people can actually ask them and say, okay, we have a proposal for this and that, and we think that it's necessary to start a standardization project on this. What I think is important to mention here is that there is the IFCC scientific division, who decides more or less eventually on whether a working group is being established. But once the decision is made, you also have to fill basically your working group. And typically working groups comprise of national metrology institutes, academia like me, I'm also involved in one of the working groups, IVD industry, who are also very important because of course they're an important link basically in the complete chain, as well as research institutes. So this is really where the work is being done, where the standardization takes place. And once there is a reference measurement system, so this whole unbroken chain of procedures and calibrators, you can submit this top of the traceability to the JCTLM, which is a Joint Committee for Traceability in Laboratory Medicine. Basically, it's a combination of, amongst others, the BIPM and the IFCC. And here, basically, there is a worldwide database of reference materials and reference measurement procedures that are in place. So that is how it works. Then we come to our current healthcare system, or should we actually call it healthcare system. Here I show you a graph of, and I do admit that it's a slightly older picture, but I think that the situation has not changed too dramatically over the time. Here I show you a graph, and what you can see is that for every drug that's on the market, for some people it is very effective, but for most of the individuals it is really not that effective. So most of the therapies really are effective only for a small subset of patients. And part of the reason for this is that we do not completely understand the underlying causes of the diseases, but also how basically the diseases progress. So we really actually need to have better diagnostics. And this is represented in this slide, where Currently, we basically have one drug that more or less fits all, and that was really the approach that I was showing you on the previous page. But what we really want to go to is to have the right drug for the right patient at the right time and in the right dose. And this basically requires us to have much more precise diagnostics and also much more precise diagnostic tests. Now, that is not to mention that we really need to have more precision in terms of what is the actual value. That is also important. But what is, I think, most important is that we actually target the biochemicals that are most clinically relevant. So that was also something that was recognized by a working group on test evaluation, headed by Rita Horvath from Australia. And she designed, or with her working group, a framework for test development. And I think what you have to appreciate in this, and what we always say is very important, is that we need to have this clinical pathway in the middle of everything that we do. We always want to ensure that what we do is clinically relevant and that it will actually benefit patients. So the first thing when we start developing a new test, or when we think of a test, is that the measurand, so the actual compound that we intend to measure, of a clinical chemistry test should reflect the clinical problem. So does it really add information on the clinical problem? Then when we start developing a test for this, the analytical performance should really be in line with the required clinical performance. And of course, the analytical performance is also where the standardization comes in. So currently, my line of research is related to proteins, and also my presentation will be focusing on proteins. But most of the clinical chemistry tests focusing on proteins at the moment are based on the principle that on the one gene, one enzyme, or the one gene, one protein principle. And that was something that we thought of in the early 1940s, when basically, we just saw we could identify individual proteins. However, we now know that this is no longer the case, because an individual human gene can actually be modified either at the genetic level or at the protein level. And through these modifications, you can actually have a large variety of proteins originating from an individual gene. And these subsets, or these variants, actually, we call proteoforms. And this is something that makes the world of protein biochemistry, I would say, a lot more complex than we envisioned in the 1940s. But I do think that, and I actually know, that these proteoforms are very important when it comes to diagnostics. And I will provide you an example in the coming slides. Moving on to antithrombin, which is my protein that I worked on from the beginning. I have a background in analytical mass spectrometry, and particularly I was looking during my PhD in protein glycosylation. And I didn't really know how to bring this basically to a patient until I moved to the clinical chemistry department. And there I said, okay, I want to have a model protein that actually has glycosylation, but not only that as interest. So what is antithrombin? It is a 58 kilodalton glycoprotein, which has an essential role in the coagulation cascade. It causes a lack of functional antithrombin deficiency, or a lack of antithrombin deficiency leads to an increased risk for venous thromboembolism, so for thrombosis. And this can either be caused by a decreased concentration of antithrombin, or it can also be caused by a dysfunctional antithrombin. And this dysfunctional antithrombin can be caused either by genetic variants or by altered glycosylation. So the current diagnostic pathway for antithrombin deficiency is that there is a suspicion of antithrombin deficiency, then an antithrombin activity test is being done, and, well, guess what? The units of the antithrombin activity tests are IU. basically it is a percentage. We have no idea how much it is or how much it isn't. And generally we say if it's over 80% it's healthy, and if it's below 80%, well, you could do an antithrombin antigen test, but most of the time, at least in the Netherlands, this is not something that's being done. In other countries, people are a little bit more rigorous in really assessing what is going on. We thought that actually this is all very nice, but when you look at the activity that is actually being measured, we think that this is a combination of the concentration of the antithrombin as well as the molecular characteristics of the antithrombin. And we said, well, both the concentration and the molecular characteristics are something that we can assess using mass spectrometry, which is why we set out to develop a test for antithrombin. Well, of course, there is a mismatch between just there has to be a real clinical problem when you start developing a test. And in the case of the antithrombin test, this is really the case, because the activity test only provides a rather vague picture of what is actually going on. For instance, if you would look at this picture here on the left-hand side, you would probably think that it is a girl with the pearl earring that you have here. However, in reality, it may actually not be the girl with the pearl earring. It may be something completely different. So this is what we really wanted to uncover and what we wanted to provide our clinicians when we started this project on the test. What I do want to mention as well is that currently there is a relatively poor diagnosis, and typically the poor diagnosis results in a poor healthcare. Often, basically, you have biological and pathological diversity, for instance, for antithrombin. Then you have the guidelines that doctors are typically referring to and that they are acting on. And because there is quite a lot of variation in the antithrombin screening test, there is quite a large difference, particularly when it comes to the identification of antithrombin deficiency. The results are often not very clear, and what that actually causes is that one, the screening guidelines are basically saying there is insufficient evidence. But the second thing is also that the therapies are a one-size-fits-all approach, because basically we just don't really know what's underneath. We just know that there may be a difference or a decreased activity, but we really don't know what's going on. And it could very well be that a different mutation in one part of antithrombin has a completely different clinical phenotype than an antithrombin deficiency caused, for instance, by glycosylation deficiency. So we really think that we have to dive deeper and we have to get a better grasp of what is going on. So when we set out, we wanted to do a molecular characterization through the monitoring of peptides. So what we do, we chop our protein of interest up in peptides through proteolytic enzymes, and then we actually measure these peptides. What we wanted to do was get basically three types of information. One, quantitative information. Two, information on the mutations. And three, information on the glycosylation of the antithrombin in the patient. So we have a method, and I do not want you to completely understand all of this, where we first capture the protein from citrate plasma. And then we do what is called the bottom-up mass spectrometry approach, where we digest the protein of the antithrombin in the presence of stable isotope-labeled peptides, and we actually measure each of these peptides. And if there is a difference in the signal for one of these peptides, we know that there actually something has changed in this peptide, and that there most likely will be the deficiency. I'm going to skip this. So we actually performed analytical validation of this test, and we assessed the precision, linearity, carryover, all kinds of things that we are obliged to do, basically, and then started to apply our test. And the first thing was, okay, there was a clinician who came to us and he said, yeah, I have this patient, and she is deficient, borderline for us. Actually, Actually, here you can see that for all of the more general tests, I would say, there is no deficiency, but for antithrombin there was constantly this value between 96 and 72, and he said, you know, this is something that is borderline, I don't really know what to do with this. She doesn't really have too many clinical symptoms, but I really don't know. So we said, okay, let's see if we can actually pick up something and if we see something. And we initially weren't very, what should I say, we didn't think too much of it, we didn't expect much. But when we actually did the measurements, we measured initially 2 peptides, which are the ones here on the left-hand side. And what you can see, wait, where's my pointer? Oh, there. What you can see immediately is that for this peptide LVSAN on the left-hand side, the values that we obtained here, but also the concentrations that we obtained here, were actually within the reference limit. But when we assessed also other peptides, we saw that there was one peptide that had about half the concentration that was expected, and it was outside of the reference range. And then we thought, okay, most likely this is where there is a deficiency, and then we actually looked into literature for what type of deficiencies we would expect, and we were also able to actually measure the affected peptide. Which you can see most on the right-hand side of the graph. So we were actually very happy, and we thought, yay, we can do this. So then we actually moved on to larger cohorts, and we first assessed quantitative defects, where we basically just look at the concentration. I have to tell you that we are actually measuring in molar units, and the reason that we're measuring in molar units is because we measure these small peptides, and they basically just refer to the number of the antithrombin proteins in a sample rather than grams per liter, because when you have glycosylation, your actually molecular weight of the protein changes, so you cannot unambiguously define the molecular weight of antithrombin and of most of the proteins. We use molar units for this. So basically we were able to identify all of the quantitative defects, and then went on to also identify qualitative defects, where we were actually able to identify this in so far, well actually now more, but at that time 34 samples to carry variant peptides. We also specifically tried to make these variant peptides visible in our mass spectrometer, and we were able to do that for most of the cases as well. So then we said, okay, we have to compare our mass spectrometry test to the classic diagnostics, and what you may appreciate is that here on the left-hand side you see the activity tests, and in general there is about 81% that is of these individuals that was classified correctly. But when we use our mass spectrometry test, we were able to identify 100%. And this is even more profound for the type 2, what they call antithrombin deficient individuals, where you really have a defect. So not a quantitative defect, but a qualitative defect. There, our mass spectrometry test really outperforms. So, of course, it is imperative that we have to basically aid the clinicians. So we kind of started to draw up a clinical pathway for this, and on the left-hand side you can see the traditional diagnostics, and on the right-hand side you see the precision diagnostics. And what we think is very important is that there is this gray zone, which is typically between 60 and 80%, where clinicians often don't know exactly what is going on, and they basically, they feel like they are somewhat guessing. And particularly for this area, which is a gray zone, we think that we could add benefit using our mass spectrometry test. Specifically because, of course, not every clinical laboratory, or maybe, of course, at least for us, of course, not every clinical laboratory has the capabilities of doing mass spectrometry. So we think that we can be, for instance, a center of excellence, and then we can move on from that. What I think that is also imperative is that we want to not only use this test for diagnostics, but eventually also want to move on into protein standardization, because I do think that mass spectrometry is the method of choice for standardization, given that you can actually identify your compound of interest at the molecular level rather than having a more broad or vague definition based on either immunoassays or, in the case of antithrombin, even activity. So these proteoforms, they're not just recognized by us. There are quite a lot of, well, not quite a lot, but there are quite some other groups that have now started to do research into the use of proteoforms for laboratory medicine. One of them is, for instance, the GFAP, but also IGF-1, and there are quite a lot of others. HbA1c is another one, where looking into proteoforms really is going to, we at least think that it's going to enhance our diagnostic potential. What I do want to mention, like I said, our research is not only based on developing novel clinical chemistry tests, but partially is also related to metrology and standardization. And I'm also part of the ProMed team, where we basically now aim to define or investigate what the influence of these proteostructures and also specifically the proteoforms is on the measurements and also on their function, and so what type of proteoforms we should consider when we really look into standardization projects for proteins. So with that, I come to my conclusions, and I think that I hope that I have convinced you that proteoform measurement is a really promising area for clinical chemistry. We can analyze proteoforms in a direct manner by mass spectrometry, and they can provide the right drug for the right patient, hopefully also at the right time. And it is important to standardize these tests, ideally upfront, or at the moment that tests are being developed, to also enable standardized cutoff values or comparable cutoff values, reference values, as well as therapeutic targets. And I think that the current efforts that are going on in protein standardization are already not relatively complicated, and generally they don't even consider the proteoform. So this is really something that I think, we will have to up our game still in order to make this happen. Thank you.
Thank you, Renée, for this insight into a very important field of medical research. I would like to now introduce our second speaker, Claude Bailat. Claude holds a master in physics from the Universität Neuenburg in Switzerland and completed a PhD in physics at the Paul Scherrer Institute. He has been a postdoctoral research associate and senior research associate at the University of Illinois and a visiting professor at Brown University in the USA. He is currently head of the radiometrology group and vice director of the Institute for Radiation Physics at Lausanne University Hospital. His research focuses on various metrological aspects of radiation physics, more specifically dosimetry, radionuclide metrology, and radiation therapy. As a radiometrologist, he oversees a calibration laboratory for the unit Becquerel and a personal dosimetry service. Dear Claude, welcome to the stage. We look forward to your presentation on bringing radiometrology to the hospital.
Hello, everybody. Thank you very much for having me. It's really an honor to be on this stage. So I will try to be more on the physics, because you saw now chemistry and the old enemy, physics and chemistry. So no, it's not true. We really respect chemists in my field. So there is just fast, we're a small country, Switzerland, but I feel that my hospital is really big. We're about 12,000 employees. And in the middle of all that, we have a little group of radiometrologists, which is normally not common in hospitals. So I will bring you two examples of practical examples of why they pay us. mostly in very short, we try to bring solutions to problems instead of problems to solution, which is what a lot of people do. And I will go in two different fields, first in radiotherapy and after in nuclear medicine. So radiotherapy, it's the kingdom of the absorbed dose to water. So it's an energy divided by a mass, it's joule per kilogram, it's the gray. And we measure that normally with ionization chambers in routine for quality assessment, for accuracy of the dose. When you will benefit from a radiotherapy treatment, you're really happy that the dose is established. But sometimes in this organized system, you can have a new technology coming. And in that case, that was what happened in the hospital in 2012 or 13. They started to do some research on ultra-high dose rate radiotherapy. And so they found with different animal models a weird effect, saying that if you increase the dose rate, normal tissue will not change much the response, but will improve the response, but tumor will not change the response. So that means that this differential effect between tumor and normal tissue is increased. So they found that with pig skin. You can see that's one of the irrigation we did. That's not really great to work with pigs, they smell. We investigated the skin. We have also kind of recognitions. It's cognitive test with mice. There's also results with fish. We're now investigating eggs. So a lot of different results. So in the system, normally in conventional radiotherapy, there is really a good system which has been set in place that I will a bit detail. And the flash radiotherapy has changed some of what was established for 50 years, and everybody believed in that. So, point A, conventional radiotherapy. First, we are in a very normative system. The linacs are normed. We know what's coming out. They have a design that allows us to know more or less the energy. We know the output, etc., etc. They had feedback loops for stability. It's really stable, well defined. There's many standards about that. The medical physicists, the people really, the physicists working in the clinics, they have all these tools. They have procedure. They know how to define a beam, how to put him in a little box so they know what it will do. They have calibration facilities. They can send it to National Metrology Institute. They calibrate the ionization chamber. And if they're not in the good place, they're not in the same beam, they have correction factors. So life is busy but pretty good. So now if you change this system like an elephant, so you end up being out of code of practice. And this is really an issue in that case. We have dose rate issue. To increase the dose rate, they pulsed the beam, which is really something for a measurement. You know, you go from cool, long measurement to microseconds, because you cannot find dose rate wherever. The field size is a lot smaller. The energy spectrum is ugly because they don't filter, otherwise they would lose some of the dose rate. So there's a lot of issues, and every single one of these issues will bring uncertainties. And we have to remember that at the end, in reference condition, we want to be about 1% uncertainty in the clinic. So yeah, this is not super easy. So how do you go around this kind of issues? So standard strategy for radiotherapy, this is something that is very common. It's kind of a process that has been established in a lot of different fields. You first have a good relative to symmetry. You anchor your system in a National Metrology Institute to bring the accuracy. And you work every day, or you have super stable machine that makes you keep this quality. So now if you're in an ultra-high dose rate, there's no absolute dosimetry. A couple of national labs, Switzerland, Germany, try to establish beams for this kind of ultra-high dose rate. That's not easy. It will still take a bit of time. So the next best thing is to use relative dosimetry and at least stay wrong always the same. And maybe one day we'll know we're wrong and we'll correct all the clinical tests. So what was done in multiple labs, so we took different dosimeters having different detecting principles. We used chemistry, we used, for example, alanine, we used thermoluminescence, we used films, which was ionization chamber. So we think that would be really unlucky that the saturation or the effect would be the same, having different detecting principles. And we move from conventional beams and we extrapolate to flash slowly. Unluckily, not in a metrology lab, in a clinical setting. So we don't have, all the tools to change the beams as we would wish. So the first conclusion for this kind of flash is you get a new technology. This is exactly what's happening every single day in imaging. Nowadays, the imaging beams are not according to the standards, because the manufacturers, they find a new way to do and they go first and think second. And our hospitals, they love technology, so they will buy it right away. But the idea is that the community brought some safety, but it's not ready for a clinical routine, for sure. I would not like to be the patient in that case. But it is more or less stable, I would say. Another part that we support a lot is nuclear medicine, because we are the designated institute for the becquerel. The becquerel is the unit of activity. 1 becquerel, it's 1 atomic disintegration per second. When you measure, for example, half-life, which is the decay half-life of a radionuclide, you very often use mass spectrometry and activity measurement, and that's a very funny way to link time and mass. And I think that's a bit unique in metrology. So there's 2 types of nuclear medicine. You have diagnostic, where you will go and have, for example, a contrast agent, radioactive, that will be injected in you and you will glow in the dark, but not really, but we'll make picture and we can see things. And there's therapy, where they will inject a lot of activity in a precise place to burn a tumor. How they do that, if we go backward, before injecting the patient, they measure, it's depending on the legal framework, but in my country, you have to measure the dose you're going to inject in a radionuclide calibrator. And this radionuclide calibrator has to be calibrated for this radio element. So you get your radioactive solution, you pump with a syringe, you measure how much activity you have, you inject the patient. You can see that a lot of the metrology is in this little radionuclide calibrator. So how do we calibrate this kind of radionuclide calibrator? For every country, there's a different way to do it, and the legal framework can be very different. But at the end of the day, the user, they want to have a dial, they put a radio element on it, and they get the activity. So how we do that? Normally you can use reference solution covering the entire energy range, or you can also use the exact same radio element. For that, we have an ionization chamber that has been calibrated for this element, which is based on a standardization. And this standardization takes, between 1 month and 10 months, depending on the radio element, to be done. And after it's compared, and that's compared at BIPM in Paris, We send an ampoule full with radioactive solution. We know how much we believe is in that. Everybody does that, and we have a very direct democratic system. The average is the reference solution, the reference value. So that's if everything works well. But we have nearly every year emerging radionuclide. And our physicians, they like to test everything that comes. And especially sometimes it's free to do the first test. And so very often you arrive and you have this new radio element and they want to use it. And the problem is sometimes we even don't have nuclear data. We don't know the half-life. The half-life is kind of important in nuclear medicine because the half-life is short. So it means between your measurement and what you inject to the patient, if you calculate wrong, this is pure bias coming in, that's an error. There's also the decay scheme. Very often we don't know them. That's something I even didn't know it was in the table of element, and you have to look where it is. They're very creative to find things. And we're not traceable, so we have to do kind of a standardization on the fly, and we have no comparison. Sometimes you have only one lab that got the solution, and so we try to collaborate not to make a big mistake and have consequences on the patient. And after, that's why I say we work a lot with radiochemists, because that's a big issue, the impurities. So very often we have to purify the solution. And that's not nice to do that. You have to do that in hot cells or with very skilled people. And the chemical behavior can be a surprise. Yeah, we have some elements which will be volatile. So that means if you keep them in a solution to inject, there's no issue. If you drop it on the table, it goes in the air, and you spend one day cleaning up the entire laboratory, and you just hope it's not a place where a patient will go through. So in nuclear medicine, it's again always a work in progress. When we think we're ready with all the radio elements, we find a new one or a new isotope. This is something that is very fancy at the moment, because you have new things coming, and the idea will be to be able to do therapy and imaging with the same kind, with a pair of radio elements. That's called theranostic, and there's a lot of money injected in that. And for us, we are just there to support clinical transfer, and after we want the system to be stable. So that was 2 examples of what we do as a metrologist, but a radiometrologist. There's not so many found metrologists, pure metrologists in the hospital. Even though, you know, we say evidence-based medicine, evidence needs measurement, and there's a lot of different measurements performed in the hospital, which have no traceability or check done on the clinical site. But it can be a pump, it can be a flow, it can be a temperature. You don't know. So this evidence-based medicine, I think we have to support the beginning of the word. I think it's difficult. We discussed with some of my colleagues, Metas, to try to enter the hospital. The chemical side is clearly, it's laboratory, and the people are in that frame. You know, the culture is there. In different places in the hospital, the culture is not there, and that's something we'll have to work on. And I have to say, if I look in the gray, now most medical physicists are super happy to have not the responsibility of accuracy. You know, you give them a certificate with a stamp on it, they're super happy. It's one less worry. It would not come in their mind to do it themselves. They love to send it to Metas, to PTB, they get a certificate, life is sweet. We could reach that system taking that as an example. I'm sure it takes time, and I think we'll need to show that we're useful, not just annoying, which is sometimes a bit difficult. So, thank you.
Thank you very much for this excellent example of applied metrology. So you all already took your chair, and for the following panel discussion, I would also like to invite Renée back to the stage, as well as Willy May and 𝐦𝐞𝐬𝐢𝐧𝐠 for a discussion on the future role of metrology in advancing technology in the life science. The discussion will be moderated by Georgette Macdonald. Georgette is the Director General of the Metrology Research Center of the National Research Council of Canada. She holds a Bachelor of Applied Science degree from the University of Ottawa and an MBA from Queen's University. Georgette is a member of the International Committee for Weights and Measures and the President of the CIPM's Consultative Committee for Mass and Related Quantities. She also serves as the Vice President of the CIPM's Forum on Metrology and Digitalization. Please join me in welcoming Georgette and the 4 panelists to the stage.
Thank you, Anna, and thank you for pulling together such a lovely panel and some wonderful talks to get us started today. I am going to start by introducing Dr. Willy May before I put him on the spot with a question. So, Willy earned his PhD in chemistry from the University of Maryland, and he started his career as a senior analyst at Oak Ridge Gaseous Diffusion Plant before joining NIST, where he led a research career in analytical chemistry for more than 20 years, publishing over 100 papers and giving more than 250 lectures. He served as the director of NIST and the Undersecretary of Commerce for Standards and Technologies, and in that role provided oversight, advancing measurement science, standards, and technology. In 2018, he took over leadership role with Morgan State University as the new Vice President for Research and Economic Development. He is also the American Association for the Advancement of Science President-elect in 2024, the President for a following year, and also a member of the Chair of the Board of Directors. Willy, I do have a question to start us off. And you can't take all the time, so that's part of that's part of the thing. So what have been some of the drivers for the development of the field of metrology in health sciences, specifically the metrology for chemistry?
Well, for us when we began, metrology and chemistry was in its infancy. So we basically had to build the framework. And I'd like to think that what you saw yesterday was birthed by some of the activities that we established with the CCQM. And again, initially we would call it chemical metrology, but I would hopefully more accurately call it applying metrological principles to chemistry and biology. And our biggest challenge was identifying a finite number of comparisons to shed light over a very, very, very broad space. In fact, one of the concepts we developed in developing our pilot studies, which actually became laboratory-based symposia, where we would invite not only the National Metrology Institutes, because in the beginning there were very few who had active programs in chemistry and biology. We would bring in the user community to participate in those comparisons. And then we would have very, very heated intellectual arguments. Some people, discussions, some people might call them arguments, but we walked out of there more enlightened, and I think friends and colleagues, to determine what the best methods were and the multiplicity of methods, if applied appropriately, could yield what we agreed on was truth. And from those studies, we would determine which techniques of measurement procedures could be used in the key comparisons. So I think that brought together a much larger community than just the NMIs, and I think that led to things like designated institutes and so forth. So I'd like to think that our activities help to expand the community in size, but also relevance.
Yeah, thank you. I think that touches on the nice point of bringing stakeholders to the conversation around, we do metrology for metrology's sake, but we also do it to help solve problems that are important to others. Excellent. I would now also like to introduce our second additional panelist to our two speakers. 𝐦𝐞𝐬𝐡𝐚𝐩𝐿 has more than 25 years experience working in the metrology field, of which most of it was in ionizing radiation. She joined the National Metrology Institute of South Africa in '97 in the electrical metrology after finishing her training and serving as a medical physicist. Then moved into the radiation dosimetry, where she worked for 11 years before joining the executive management team, where she was responsible for the division of ionizing radiation, which later combined with the chemistry section. She served as an executive at NMISA for 8 years before joining the International Atomic Energy Agency in 2020 as a medical radiation physicist. Her expertise also covers legal metrology and accreditation, as she served as a member of the Metrology Advisory Committee for more than 5 years and worked with the South African National Accreditation System as a technical assessor in the field of ionizing radiation. Welcome. So, can you please describe for us the role of IAEA as an international organization in fighting cancer?
Thank you for this opportunity and for the invitation. The IAEA has been working with member states for more than 65 years in the area of tackling cancer through nuclear applications. We do quite a lot of activities, from training of personnel that are supposed to be delivering the services to the hospital, in the hospitals, to also helping the member states build the facilities that are needed for treatment of cancer. And also we write basic safety standards which are adopted by member states as regulations that actually govern the use of nuclear applications. And in that also it explicitly states the requirements for calibration and the required specialist that needs to be in the clinic for them to implement some of the nuclear applications, especially in radiation therapy and nuclear medicine and diagnostic radiology. In those safety standards, it also gives the responsibility to member states or to the license holders to have audit programs. You will see later on why I'm actually bringing this out. So not only in the space of guidance documents, we also, as regulations, we also write guidance documents, which was mentioned earlier on, that guides the professionals on how to actually link measurement traceability from the primary standards to their equipment in the clinic. And we have an IAEA WHO SSDL network, which actually calibrates most of the user equipment that are used, which primary standards would not necessarily see. And through this network, we also have various activities where we train the radiation metrologists on how to actually perform calibrations and also how to support the medical physicists in the clinic. So it doesn't just stop at calibration, they need to be able to support the users in actually ensuring the dosimetry. We also have what we call coordinated research projects, where we bring in different member states from low and middle income countries and from developed economies and bring them together to bring solutions. So our approach is not only from the physics perspective, we also have clinicians that we work with that are at the IEA, and they run coordinated research projects looking at some of the specific procedures that might be of benefit to the community in terms of maybe shortening the treatment times and just ensuring that cancer treatment is accessible for all and is as comfortable as possible, ensuring that we still deliver the dose that needs to be delivered to get the best outcomes from the treatment. In 2020, we launched an initiative called Rays of Hope. Through the Rays of Hope, it was really intended to assist the member states because there's still countries that do not have any capabilities for radiation therapy. So it's assisting member states to actually bridge the gap and establish the facilities. We work with international organizations like WHO in assisting the countries to achieve those results. And in some aspects of what we do, we also connect what we've done within metrology. We have what we call 𝐦𝐞𝐭𝐫𝐬 centers, which are centers that have developed and have techniques that they can train some of the people in their regions. So in each region, we're trying to establish some of the 𝐦𝐞𝐭𝐫𝐬 centers, which will have an impact both from the clinical perspective as they train the clinicians to the metrologist or medical physicist. So that's in short what we do.
Thank you. I think that it's really helpful when we talk about what we do as a community of metrologists to see how it impacts the other organizations and the other international organizations that we partner with or collaborate with in terms of impacting their communities as well. This is very helpful to remind us. Renée, I'm going to ask you if you can speak a little bit about the role metrology plays for the validation of data for personalized medicine.
Yeah, so thank you for this question. I think that already during my presentation, I pointed out that for clinical chemistry or laboratory medicine, it is very important that results that are being generated are transferable between the different laboratories, particularly because clinicians rely on clinical cutoff values and treatment targets. And I think that it is, in order to have a sustainable healthcare system, very, very important that we do not have to redo these, basically the reference values and also other studies over and over again for a different laboratory or for a different IVD manufacturer in order to have clinically actionable results and actually eventually also the best care. And it's for a clinician, it is very difficult to realize that, you know, if they move to a different hospital because, well, they just transfer, they change jobs, that they have to deal with different reference values just because their laboratory has gone over to a different IVD manufacturer. And I think that is really where we need the metrologist, and this is something that I think the clinical chemistry community is aware of, or at least part of them are aware of. But like in Lausanne, it's not something, there are not many metrologists in the hospital, and it's also not a very a rewarding task to do, I would say.
Thank you, which actually does lead me back to the next question is really, and you did touch on it in your presentation, but what are some of the challenges of bringing metrological traceability into the hospital world?
In my field, I always say that everything can be solved, you just need time and money. And unluckily, we have issues to follow the medical field is going so fast, because it's true, it's an investment place, and everybody is getting older, and we all want to be cured of all the diseases we have. And so it means there's a lot of money, the research is going fast, but the metrology is not well funded. And so that means we're always lagging behind, trying to catch up, and we're just reactive. And unluckily, we also need to go back to places where a lot of countries stopped to study. For my country, it's like radiochemistry. It's been, I think we stopped training radiochemists in Switzerland maybe 15 years ago. And so we have a lack of luckily we're in the middle of Europe, so we steal them from all the neighboring countries. But for me, it's really, we have issues to be proactive, and the system goes too fast for a poor radiometrologist.
𝐦𝐞𝐭𝐫𝐤, perhaps I will follow on the what role do you think ionizing radiation in cancer treatment can continue to expand in the future, and how is that interplay between the metrology laboratories and the IAEA?
There's still quite a big role. I mean, we see the technology development from diagnosis to treatment, as my colleague says, it's moving so fast because of the need from the clinicians and from the patients. But the gap, the challenge is that metrology is running behind and running very fast to try and catch up. Not that it's not, there's nothing being done, but I think there's room for industry to work together with metrology institutes to try and bring the solutions together. Otherwise, we will never be able to catch up. It's really going to take the working together to bring solutions, even if maybe I guess maybe the industry might not be as open as in quantum, but there needs to be something that brings industry and metrology together to bring better solutions. And we've seen in some applications that because we're lagging behind, there has been incidences that have been quite bad, but somehow this needs to be closed up. Just maybe for metrology, I mean, metrology has really done quite well in terms of the development is not really on the standards need, it's on the user need. So the measurement solutions more than the development of standards. So and metrology institutes have been really trying hard with the budget cuts and everything to try and fill up that gap of actually bringing a solution to the user community.
And so, Willie, maybe I can follow on that one. Is where do you see the role of CCQM and the CIPM and the BIPM in terms of addressing those gaps and making the conversations with industry and stakeholders?
Well, that role is continuing to evolve. And I think you saw some of that evolution in the presentations we had from the young folks yesterday. That is trying to work with the user community to develop a number of comparisons, a study, to cover a very, very broad space, to add to provide some level of of comparability over both time and space in this very, very broad area. That is bringing different voices to the community, if you will, not only the National Metrology Institutes, but some of the customer labs also. Obviously, they are not a part of MRA, but having dialogues with them, because the whole idea is that we should be providing a framework for them to support comparability of measurements over time and space. And obviously, in the biology area, that is a lot more difficult than it is in chemistry, and I would say in physics as well.
Thank you. All right, well, with that, I do get the final word, and the final word is thank you so much for bringing this conversation to the table. The idea of what future revolutions for metrology in life sciences might look like is actually much bigger and broader than we had the opportunity to touch on here, but your specific examples and your contributions allow us to have a very concrete discussion about things that we might need to do in our own institutes. And so for that, I thank you very much for your contributions.